Today we'll be talking about the reactivity-informed pharmacophore editing and biological evaluation of andrographolide and the synthesis of some A-ring analogs, specifically closing the loop on the Oxetane. Next slide. So as a quick brief, I'll start off by introducing the compound of interest, andrographolide, then talk about the library of C19 analogs that we've synthesized, and in addition to their biological evaluation. And finally, we'll talk about our current efforts, or actually the methods through which we have constructed the Oxetane moiety on andrographolide, in addition to our current efforts towards some A-ring elaboration. Next, so andrographolide is a natural product that is isolated from the plant Andrographis paniculata. This plant is located in the Asian region and has been used for numerous years in Ayurvedic medicine, specifically to help treat diseases of liver, skin diseases, and fever. And this compound in particular, andrographolide, is known to have some anti-cancer and antibacterial properties, and it has also been studied in various clinical trials in the past, but hasn't really pulled through due to like metabolism and bioavailability issues. Andrographolide is known to primarily exert its anti-cancer capabilities through the inhibition of NF-kappa B, which is a transcription factor that modulates various genes relating to cell survival and metastasis. But actually, to step back a bit, much of the inspiration in our laboratory to actually work with natural products comes from the statistic that about a quarter of FDA-approved drugs are either natural products themselves or derivatives of natural products. And on the right, you can actually see some of the current therapeutics that are inspired by natural products. So we believe that there is clearly much benefit to taking a natural product and making key modifications with the goal to make them more potent and provide, I guess, much more medicinal value. Next, and so relating to andrographolide itself, the inspiration for our study came from the study from a group in Thailand who had reported these two analogs, the C19 trityl and the TBDPS epoxy andrographolide analog. And interestingly enough, they had found that these two analogs actually inhibit the WNT 𝜷-catenin signaling pathway, which is essentially a biological pathway that has to do with cell proliferation and differentiation. And this pathway has some very many key kinases, such as GSK3 beta, which essentially phosphorylate 𝜷-catenin and tag it for degradation. Specifically, the TBDPS epoxy andrographolide analog inhibits GSK3 beta, basically preventing this phosphorylation and preventing its entry into the nucleus. And notably, the trityl compound inhibits this WNT 𝜷-catenin pathway independent of GSK3 beta. And we found this very interesting as it allowed us to ask this question of whether or how large hydrophobic substitutions at the C19 position changes the biological mechanism of action through which these compounds exert or act through. And so here's a library of the 12 compounds that we synthesized, including the trityl and silyl ether analogs. And in terms of synthesis, most of these compounds followed a simple protection conditions. Next. And so with these compounds in hand, we wanted to test their ability or their antiproliferative activity in different cancer cell lines. And so we picked four cell lines in particular, HCT116 and HT29 colorectal cancer cells, and MCF7 and MDA-MB231 breast cancer cells. And so we performed this experiment at 24 and 96-hour time points. And I guess just to pull out or highlight some few points or takeaways from this data or these many numbers, we found that generally our compounds were much more potent than the parent compound andrographolide. And compound 13 in particular displayed, I guess, 1 to 2 orders of magnitude increased potency than the parent compound. And importantly, actually, all of our compounds generally had lower IC50 values in HT29 and HCT116 colorectal cancer cells, as opposed to the breast cancer cells across both these time courses, showing that there is some cell line selectivity here. Next. And so in order to evaluate or investigate the ability of our compounds to inhibit NF-kappa B, we performed a secreted alkaline phosphatase assay, or a SEEP assay. Essentially, the addition of tumor necrosis factor alpha, or TNF-alpha, leads to the activation of NF-kappa B. And the translocation and binding of NF-kappa B to the promoter can then be measured spectrophotometrically by the activity of the resulting SEEP on a colorimetric substrate called quanti-blue. We ran a similar experiment on a CD40 cells with the pathway activation by CD40, the CD40L ligand. And notably, amongst both these experiments, the trityl ether analogs reduced CEAP activity in a relatively dose-dependent fashion in these HEC-293 cells. And compound 13 in particular, the chlorobutyl compound, displayed the greatest dose-dependent decrease in both these experiments, which was quite interesting. Then, in order to evaluate our compound's effects on Wnt1 signaling, we used an in vitro reporter cell experiment. We used a stably transfected Wnt1 responsive 3T3 cell line, which expresses a TCF-LEF activated, basically a firefly luciferase reporter gene. And we activated this Wnt1 pathway using Wnt3A, and then dosed our compounds at 10 and 5 micromolar. After the treatment of these compounds in these cells for about 24 hours, and with the addition of a lysis luciferin substrate solution, we observed the luminescence. It was also previously recognized, and I had previously mentioned, that some C19 analogs show GSK3-beta dependent Wnt1 modulation or modulation of this Wnt1 pathway. And in order to actually assess whether the activity of our compounds in this Wnt1 pathway might be altered by direct inhibition of GSK3-beta, we co-dosed our compounds in the same reporter cell line with 10 micromolar of CHR, or CHR99021, which is essentially a selective GSK3-beta kinase inhibitor. And that was done to activate the luciferase expression, essentially. And so what is consistent within both experiments is that the trityl ether analogs exhibited kind of a potent inhibition of luciferase expression. Next. We then investigated whether the administration of these compounds, whether it changes the phosphorylation or localization of beta-catenin in those specific cell types. In order to do this, we stained beta-catenin with an primary antibody followed by a secondary fluorophore antibody. And then we counterstained with DAPI to highlight the nucleus. And we used ImageShade to attempt to quantify the percent overlap, or the percentage of activated and non-activated beta-catenin in the nucleus versus the cytosol. And overall, like, the data regarding this overlap or difference between the DAPI stained nucleus and the cytosol was kind of insignificant, allowing us to conclude that our compounds do not cause a significant change in the beta-catenin distribution, and the difference in the absolute beta-catenin content is minimal upon dosing of our compounds. And so overall, we can suggest that even though beta-catenin is involved in the function of these analogs, the full mechanism definitely involves many more factors just beyond one target. But we can kind of confidently say, based on this in vitro data, that subtle changes to that C19 position, or the A ring of andrographolide, can have great consequences on the potency and mechanism or biological mechanism action of these compounds. Next. And so yeah, this part of the study is actually published in Bioorganic Medchem Letters. But yeah, next. The next part of our study actually involves synthesizing this material, which is isolated from a small portion of the Andrographis paniculata plant. This material contains the oxygen moiety, which is highlighted in blue on the right.
My mind would get gas. Again, aside from the fact that the hardware is already
and its biological activity has not been studied in the past, considering that it is quite challenging to construct in itself. But you might be wondering why this small portion, or this small percent of this Andrographis plant, why it attracts our attention. This Oxetane moiety, which are actually bioisosteres of carbonyls, and these functionalities are actually metabolic liabilities as they react with biological amines. But oxetanes, which have conformational kind of a rigidity, have a hybridization that is close to sp2 and are basically not as reactive. And on the left actually shows some examples of natural products that contain this oxetane moiety. And in fact, it has been shown that on Taxol, or the oxetane that is on Taxol, plays a major role in its biological activity.
Okay, so in terms of our retrosynthesis for actually constructing this oxetane, we envisioned two different bond disconnections. and both of which would involve installing a leaving group at the C3 and the C19 positions respectively, and then closing the ring with an SN2. So looking at the conformational, taking a conformational analysis of the Andrographolide, we actually noticed that there's a very significant thermodynamic barrier into construction of the Oxetane ring, because as you can see in this bottom figure, construction of the Oxetane leads to a more unfavorable conformation of the ring, where it turns from a boat structure into, where it turns from a chair structure into a boat structure. Yeah, next please. As I mentioned earlier, our attempts involved construction or functionalization of the A-ring at the C3 position. So our initial attempts started with protection of the C19 alcohol with a silyl ether group. Here is the TBS group. And then we used very harsh conditions such as PBr3 in order to functionalize the position and invert the stereochemistry so that there would be the correct trajectory into closing the ring. However, most of these conditions proved to be far too harsh for the substrate and led to decomposition. To alleviate these problems, we then developed this pathway to protect the reactive C14 alcohol on the top with this procedure that involved first acetonide protection of the C3, then C19 alcohols, then silation at C14, and then subsequent deprotection to give us the silated andrographolide intermediate with high yield and with only one chromatography. However, we later found that there would be issues deprotecting this extremely stable TIPS group using the traditional procedure of just TBAF, which led to a sort of side study. In this study, we looked for various other groups at the C14 position, similar to TIPS, but much more labile, in the hopes that we would be able to deprotect it much more conveniently than TIPS group. So we attempted to attach on a DMT silyl ether, the TBS, and the TES silyl ethers onto the C19 position. However, we found that when trying to deprotect the acetonide group at the end of the process, we found that sometimes these conditions would cleave off the silyl ether as well, which led to our findings that using the HCl salt of triethylamine would selectively cleave the acetonide in the presence of the TBS silyl ether, giving us the final product. However, this study would later be abandoned because we found a much better procedure for cleaving the TIPS group. So with this TIPS intermediate in hand, we then looked for ways to functionalize the more accessible primary alcohol at the C19 position. So one of our first attempts at doing this was using the Appel reaction, which would be able to halogenate the C19 position. However, we found that because of the reactivity of andrographolide, some of the intermediates in this reaction and some of the byproducts in this reaction would be able to intercept the substrate and lead to unwanted side reactions, such as the one on the bottom, where the bromoform conjugate base would be able to add to the alpha beta unsaturated ketone over here and lead to this undesired byproduct.
So next, we tried to minimize this this byproduct by using CCl4 instead of CBr4. However, while we did find that it resulted in less of the side product, we found that upon isolation of the C19 chloro, it did not form the oxygen immediately, and in fact, you actually had to isolate it and introduce a strong base such as sodium hydride in order to close the oxygen ring. However, upon addition of these bases, we found that chlorine was simply just not a good enough leaving group to form the oxetane. This resulted in us trying to install different other leaving groups that are much stronger, using a variety of anhydrides such as triplet anhydride or trifluoroacetic anhydride. So we start with the same C14 TIPS protected andrographolide. However, we found that through a variety of experiments that we first tried triflic anhydride, which we found only results in the conversion to the oxetane at negative 78 degrees. However, we found that this is extremely sensitive, and that upon taking the reaction out of negative 78, everything decomposes, making isolation of the oxetane very difficult. The next thing we tried was Sokal 2, which we were able to actually form the oxetane very nicely, however, at very low yields, around 33%, which made us still want to explore other options, such as TFAA, which we found if you actually start the reaction at negative 78 and slowly let it go up to room temperature, we get up to 68% yield, which is 2 times the amount as Sokal. Additionally, well, as Galen previously mentioned, we found a different way to deprotect the tips with TBAF, where we found that upon temperature control, where we started at negative 78 and then let it go up to room temperature, we get very nice conversion to the C14 deprotected andrographite and just left with oxetane andrographite. Next, we were kind of interested in mechanistically understanding why different leaving groups were actually better at forming the oxetane compared to others. So we did some DFT and Monte Carlo simulations. So first, we took the andrographite tips material with different C19 leaving groups attached and put it through Monte Carlo simulations so that we get the most conformational, the best low energy conformational structure where the C3 can perform an SN2 on the C19 carbon. And then we did further DFT optimization to get the lowest energy conformation. And from these, we measured the distance between the C3 alcohol and the C19 carbon, where a lower distance between those two groups would mean that is more predisposed to do an SN2. And what we found was that it actually very nicely correlated with our experimental results. We found with the TFA leaving group actually had the lowest distance, where if you can remember, also had the highest yield, followed by the chlorosulfite with installation by the Sokal 2 and then by a triflate, while those other leaving groups that we found didn't work, such as the mesylate and chloride, had very similar distances compared to just regular andrographolide, which of course in itself cannot make the oxetane. Additionally, we also want to pursue other C3 functionalization to form the oxetane because we theorized that different intermediates that can be intercepted may be biologically interesting in itself, such as the C3 ketone. So initial attempts at actually reversing the stereochemistry so that C19 can do an SN2 led to very poor conversion. Especially this entry 7, we initially thought there was conversion. However, upon analysis with NMR and LCMS, we formed a different product that we did not want. So what we instead decided to do was we decided to take a different approach to inverting the stereochemistry. First, we protected C19 with the TBS, then did an oxidation, where we then deprotected TBS again to do a stereoselective reduction, such as an Evans-Saxena, with some bulky borohydride. However, we have difficulties doing this. And so we're trying to pursue other methods such as a Naori hydrogenation. So current efforts towards more avian elaboration is first forming this dibromo DNP, which we can then input zinc into the reaction and form this cyclopropane acetonide. However, we have a difficult time actually installing a dibromo acetonide, where we found that actually 0.05 equivalences of CSA and THF is the best so far, but it still performs very sluggishly. So we want to do additional experiments such as using 5 molecular sieves in order to absorb the methanol byproducts and force equilibrium. Additionally, we want to form the C19 ether, where we found actually through test reactions that we can install with a methyl mesylate and some strong base. And additionally, we want to do some selective oxidation where we can selectively oxidize C19 without also oxidizing C3. And so we tried a variety of oxidation conditions such as tempo bleach, DMP, and TPAP NMO. However, we found that TPAP NMO's exclusively gives us the C19. However, we want to scale up in order to fully characterize with NMR. Additionally, we want to deoxygenate C19 or C3 or both. And we theorize we can do this with a reaction called a Bodeman combi, which uses carbon disulfide, a strong base, methyl iodide, followed by AIBN, and Bu3SNH, which we can. However, we also think that another Boeing catalyst may also work. Additionally, I mentioned before, we want to do a nitroid hydrogenation. However, that requires us turning the C19 alcohol to a C19 ester. However, this is very difficult, and we are still exploring this route. And if you want to learn more, here are some QR codes to the different publications on this project. We'd like to thank our advisor, Dr. New, and other group members, Tiffany Gu and Ali Kong, Krina Zhao, Songhyuk Ko, Selena Z, and Yeonho No, and our collaborators at ASDIP. Thank you.